So as I mentioned, you need really two important parts of the enzyme: the binding site and the catalytic site. The binding site is a larger, if you want, pocket or cleft where the substrate is situated in. Typically, that's an example of a domain. Typically, that may require about 100 amino acids. But remember, this is part of the tertiary structure. So these amino acids may be from different parts of the protein after it has folded. They line the pocket where the substrate is going to fit in. Importantly, the substrate and the enzyme will interact through non-covalent interactions of the substrate with the R groups that are lining up that binding site. Okay? No covalent bonds, just interactions between the substrate and the R groups that are exposed in the binding site. Within the binding site, there may be a very small number of amino acid residues that are involved in catalysis. That means they are actually going to be part of changing the substrate to product. So this is much smaller site. This is more in terms of situating the substrate in the pocket in the right way so that the enzyme can work on it. So I just covered that, and really most of the interaction, all of the interaction is going to be non-covalent between the R groups of the binding site and the substrate. The old model was a lock and key model. We don't really accept that. It's no longer accepted. So the new model is more of a sort of dynamic induced fit model, meaning here's the enzyme and here's the specialized pocket. It's more dynamic rather than rigid. It means it can sort of change its shape and accommodate the substrate so the fit to fit it that way. So that's the more likely that the binding site has a dynamic characteristic. It can change its conformation to accommodate the substrate.